I know, this is a challenge, isn't it, for the bladder and mind, both. Okay, so this is going to be the pyruvate dehydrogenase, and we're finally going to start getting into the citric acid cycle. So this is the citric acid cycle. You can see that wonderful cyclic shape to it. So the citric acid cycle is going to be what harvests your high energy electrons. Citric acid cycle, oftentimes denoted as being CAC. This is going to be the series of oxidation reduction reactions that's going to result in the oxidation of an acetyl group to 2 molecules of CO2. This is typically considered the final pathway for the oxidation of fuel molecules. Your oxidation is going to generate your high energy electrons. This is what actually powers your ATP synthesis. Though keep in mind, this and powering the ATP synthesis, these things are interlinked, okay? It's not like one happens, then the other one happens like minutes later or something, okay? The important sources for your precursors are going to be for biosynthesis. So when you're talking about all your other biosynthetic pathways, they all typically sort of shoot off of the citric acid cycle. We've seen a little bit of this so far, but this is where this is really going to become important, and we're going to start tying a bunch of these pathways into each other, right? They're all going to get intertwined in these citric acid cycle intermediates. This is also called the tricarboxylic acid cycle, TCA cycle, or Krebs cycle. And so this has 3 names, which is just all sorts of fun, and I totally blame the biologists for it. I totally blame the biologists for it. I will not take the credit for that one. This is the acetyl-CoA right here. You can recognize the acetyl group in red here. And this is the coenzyme A right here. And that's that file we had talked about just a bit ago. So most fuel molecules are going to enter your citric acid cycle as your acetyl-CoA. This is that one that was right after pyruvate. Remember? Pyruvate to acetyl-CoA into the citric acid cycle. Right? And that's the majority. The pyruvate dehydrogenase complex in your citric acid cycle. So your pyruvate dehydrogenase and complex, this is a large enzyme complex. This is going to oxidatively decarboxylate pyruvate to the acetyl-CoA. This is done under aerobic conditions. The acetyl-CoA is going to enter your citric acid cycle, where all remaining carbons are going to completely oxidize to the CO2. The reactions for your pyruvate dehydrogenase complex in the citric acid cycle are actually going to take place in your mitochondrial matrix. So remember that this is going to be back in that mitochondrial matrix, and we already got hints of that when we were looking at the gluconeogenesis and going from there. Okay? So again, this is your mitochondria. Ooh, so beautiful. Did you know that mitochondria is directly related to your to mental disorders? Yeah, the majority of mental disorders are actually can be predicted by mitochondria and their activities. I know, right? And so even though it's called the powerhouse of the cell, a better name for it would have actually been precursor house. It's the fuel and byproduct shipping container that everything has. So this right here is the matrix. It's the innermost part of your mitochondria. Around that, we have the inner mitochondrial membrane. This has 2 membranes that enclose this space. Okay, this is not actually membrane inside this entire area. Okay, hopefully you all kind of knew that already. Yeah. Okay, and then we have the outer mitochondrial membrane. This red one being the inner mitochondrial membrane. This is what it looks like when you actually take a picture of it. Hey. Okay. Anybody know where you get your mitochondria from? They're from your mommy. Yep. And they're highly conserved. They change very slowly. Okay, so this is an overview of the citric acid cycle. And we're sort of going to sand down and get more and more depth as we go. So again, your citric acid cycle is going to remove electrons from your acetyl-CoA, which are entering right here on the top. And it's going to use these electrons to reduce NAD plus and FAD to the forms of FADH and FADH2. Now, keep in mind, when we're looking at this citric acid cycle, we're looking at one side of the machine, okay? These FADHs, these NADHs, these things are actually better thought of as little crankshafts or little gears, okay? And they're going to feed back in to the electron transport chain, okay? So one of the ways you can look at them is this right here. If this was a turbine, this would be the head part that the water is moving, okay? Whereas the other part is going to be the part that we're shunting the electricity to, and then it's affixing the energy so we can ship it. Does that make sense? Okay. So the electron transport chain, this is the other half of that. This is going to be a series of membrane proteins. The electrons are going to be released in the reoxidation of the NADH and FADH2. This is going to flow through to generate a proton gradient. This proton gradient is going to build up, and then this is going to push through that across the inner mitochondrial membrane. The protons will flow through the ATP synthase to generate the ATP from the ADP and the inorganic phosphate. And so this is what this kind of looks like in a two-dimensional representation, okay? So here we have the citric acid cycle. Here we have the oxidative phosphorylation. Remember, though, that these things are joined, okay? Their joints are the NADH and FADH2. So we're going to get fatty acids, like glucose, and amino acids that are all going to go down to the acetyl-CoA. Now, I know I said that the fatty acids cannot generate glucose before, but I was talking about plugging into the glycolytic pathways, right? The, gluconeogenesis pathway, technically. Yes, you could take acetyl-CoA and come back through into gluconeogenesis through pyruvate, right? But that is not as energetically effective. So anyways, this right here is going to be the acetyl-CoA. This is where we typically call this as the beginning. And again, that's the beginning because fatty acids come in here, amino acids and glucose. They all join at this point, okay? Okay, so the acetyl-CoA, this is going to go through your citric acid cycle. We're going to get 2 CO2s and ATP. And then we're going to generate the NADHs and FADH2. These are actually just moving electrons. Remember, those are the electron transporters. Okay. This electron transportation is actually going to facilitate the moving of H+ from the matrix into the inner mitochondrial membrane space. Once we've transported approximately 36 H+, depending on the organism and the person. Less H+ means you're more efficient. More H+ need to mean transport means you're less efficient, in case you're curious. This is then going to give you the proton gradient density, the charge gradient density across that membrane that you need to push that ATP synthase right here to make ADP to ATP. So we assemble the ADP and a free phosphate to the ATP. By the way, this is where we're using that oxygen. Notice the oxygen is not actually directly responsible for the CO2. It's a bit different than burning fuel normally, right? We've actually split this up more like a fuel cell. Anybody ever heard of a fuel cell before? Yeah? No? Kind of? Yeah. So we've split this process up. We've converted the carbon to carbon dioxide and the use of the O2 to water. And they're separated spatially and temporarily. The pyruvate dehydrogenase complex is going to be highly integrated. It's going to be a unit of 3 distinct enzymes in your mitochondrial matrix. We have the oxidatively decarboxylates the pyruvate to acetyl-CoA. And so this is that reaction right here. We have pyruvate going to CoA. We have our NAD plus going to NADH and NH plus. Notice this actually produces a CO2 as well. The reaction is going to be catalyzed by the pyruvate dehydrogenase complex. It's an irreversible link between glycolysis and your citric acid cycle. What's important about CO2, by the way? Is CO2 charged? No. Is it soluble? To a degree. Most importantly, can it move past membranes? Yeah. So this step right here, the acetyl-CoA, that's trapped inside our mitochondria, right? But the CO2, that is not trapped, is it? So if we have a high concentration of CO2, it's just going to diffuse out of our mitochondria, isn't it? And so this is an irreversible step. It traps this reaction and it makes it go forward. Okay? Because you're going to continuously lose that CO2 and you can't go backwards. So the reaction is going to be catalyzed again. That's that irreversible link between your glycolysis right here, the pyruvate. Once you're at pyruvate, you could have went backwards. Okay? And on the other hand, the acetyl-CoA typically does not go backwards because getting this CO2 back in here and getting these electrons back is going to be very, very difficult. Okay? Getting a concentration of CO2 to go backwards in this reaction would be basically impossible. Okay, so your citric acid cycle, again, is going to be entered by the acetyl-CoA. So the pyruvate dehydrogenase complex of your E. coli, in this case, is the pyruvate dehydrogenase component. This we call E1. The dihydrolipoyl transacetylase, which is E2, and the dihydrolipoyl dehydrogenase, which is E3. The prosthetic group on each of these is E1 is TPP, E2 is lipoamide, and E3 is FAD. Remember those prosthetic groups that we have to attach to those for them to have function. The reaction that we actually catalyze with each of these portions is the oxidative decarboxylation of the pyruvate, that's the E1. The transfer of the acetyl group to the CoA, that's the E2. And the regeneration of the oxidation form of the lipoamide, which was that prosthetic group up here, that is the E3. By the way, note that the oxidation has an FAD, that's the prosthetic group for it. That was your big hint. Okay, that's a oxidation going on. The catalytic cofactors are going to be thymine pyrophosphate, which is TPP, lipoic acid, and FAD. The stoichiometric cofactors, these are going to be the cofactors that will function as substrates, are going to be your CoA and your NAD+. This right here is your TPP. Again, that's thiamine right there, pyrophosphate. This right here is lipoic acid, right there. That's the acid part. Okay, so the steps that have to be coupled because the free energy of decarboxylation step is going to drive your formation of NADH and acetyl-CoA. So here we have the pyruvate. This is going to be the decarboxylation. We're removing a CO2. And we're going to be left with this portion right here. This is then going to have the oxidation. And so we're removing 2 electrons, which will give us the positive charge instead of the negative charge. And then finally, we'll have the transfer to the CoA. And so we transfer this to the CoA here. That positive charge facilitated that reaction onto this thiol right here. All right. And by the way, you can kind of tell why these all need to be coupled together. Most of those steps are not stable at all, are they? No. Carbanion, carbocation, right? If you release those into the environment at all, allowed them access to water, anything, they would have reacted, wouldn't they? So the decarboxylation step, this is going to be the pyruvate. It'll combine with the coenzyme TPP. It'll be decarboxylated to yield hydroxyethyl TPP. The rate-limiting step in the acetyl-CoA synthesis, this is the one that does not have a super high energy intermediate. And so it's the only one the activation energy is actually significantly larger than the, in this case, the reactant to the substrate, right? And so it's the slow step. This is going to take pyruvate, the carbanion of TPP. It's catalyzed by the pyruvate dehydrogenase, but component E1. Because remember, this is a multi-enzyme complex. That whole enzyme is this. And then we have E1, E2, E3 that we're going to be doing. And again, the TPP here, which is forming that carbanion here, that's that prosthetic group. And we'll have to reset it later. The hydroxyethyl TPP is going to be that complex we form. This is actually where we lose the CO2. After this step, that loss of CO2 actually helps this keep going, though at this point we're such high energy because of this charge here that it goes forward quite rapidly. The step 1, the carbon center of your TPP is going to ionize to form carbanion. Step 2 is the carbanion is going to readily add to the carbonyl group of your pyruvate. Step 3 is going to be decarboxylation. By the way, does this remind you of anything? Remember the reactions we did at the end of organic chemistry, where we had a negatively charged group, a nucleophile, attack carbonyl. It would transfer electrons up, come back down, and then we lose a leaving group. Yeah, it's this. Your biology is just doing it. So your mechanism of E1 here, this is the complete mechanism. Does anybody have any questions about this? This is that same sort of carbanion attacking a carbonyl, going up on here, coming back down. You could go either way at this point, but if you go towards the carboxylate, losing the electrons, then you'll lose the CO2. Okay? And which is pretty likely to happen because CO2 is quite stable and will leave and won't be able to come back on, right? This will give you the resonance forms here. This is resonance stabilized, which is why this is more stable and much more likely to happen. And this is your hydroxyethyl TPP that we ended up with step 1, the first part of this E1. Okay. This is going to be the oxidation step. This is the lipoamide. This is going to be a derivative of lipoic acid. This is going to be linked to your side chain of a residue by a amide linkage. A hydroxyethyl group is going to be attached to your TPP. This is going to oxidize to form an acetyl group while being simultaneously transferred to the lipoamide. It'll yield the acetyl lipoamide. It's going to form an energy-rich thioester bond. That's the thioester right there, by the way. The disulfide group of lipoamide is actually reduced. And this is the reduced disulfide. Here. There was the reduced. And this is going to be catalyzed by your E1 again. And so right here we have that product from that first part. Here's the lipoamide. Again, it had that disulfide linkage. We're going to have this break. This right here would have had a sulfur to sulfur bond. It taking on a hydrogen and bonding to a carbon is a reduction. We're gaining more electron density on those. And this is relatively energetically favorable. This will regenerate the carbanion of your TPP. Right there. And this will give us our acetyl lipoamide. Okay, is it time? Fantastic. Let's call that a day. I will see, well, I guess all of you.